Hello, good afternoon. Welcome to the NAUMD webinar. Today's topic, very excited about filling the gap in worker safety, the role of protection from microbes in uniforms and image apparel. And very excited to have both Ben Favret, the founder of Vestex, Protects here today, as well as Dr. Matthew Hardwick, who's president over at Resinnova Laboratories. And a quick plug, if I can, for the NAUMD. We are a trade association for North American uniform manufacturers and distributors. We have news that's distributed weekly and regularly. You can opt in at naumd.com. We produce industry data and reports, educational activities such as these webinars and podcasts. And then we have our events where you can network. We have an annual trade show that we certainly encourage everyone to come to. This year it's in San Diego, October 24 to 26 at Paradise Point. And please see the website again for more info on that. That is naumd.com. And with that quick plug, Ben, I'm going to turn the webinar over to you. And thank you for you and Matthew's participation today.
Thank you, Rick, and great selection for a place to do your welcome back from pandemic trade show in San Diego. What a great city that is, and a fun place to go to for sure. Thank you all for joining us today. As I said, my name is Ben Favret. I'm the founder and CEO of Vestex. And there's no denying that the world we live in has dramatically changed over the last 18 months. The concern about worker safety and infection prevention is higher than ever in the C-suite. This Deloitte study really drives the point home that healthcare issues, disease prevention is now a top 5 issue in the C-suite. And it's also a priority, not just in the C-suite, but also for the workers on the front lines. They're worried about their safety. They're worried about their families at home and what they may bring home from work. So the new world requires new strategies to fill these gaps we have in worker safety and protection from infectious disease. As I said, I'm Ben Favret. We founded Vestex in 2009 and have been developing Vestex and doing all the research for Vestex over the last few years. And today we want to talk about 3 things. One is define the workers at greatest risk, the gaps that we have in how we address healthcare worker apparel and protection. And then the last thing is some gaps in the testing methods in ways that we go about testing. And to have that discussion with us, I'm honored to have Dr. Matthew Hardwick, the president, founder, CEO of Resinnova Labs with us. Matt is truly one of the thought leaders in infection prevention, environmental health and safety, as well as antimicrobial surfaces and textiles. He has 20 years of academic experience at Georgetown and Johns Hopkins. And his most recent academic appointment was at MedStar in DC, where he did research specifically on healthcare environments and antimicrobial apparel and textiles in the clinical environment. Matt's widely published, has presented at AATCC, ASTM, all the big meetings, and super excited to hear what Dr. Hardwick has to say today. Matt, welcome.
Thanks so much, Ben. I'm looking at this picture and a lot has changed since that picture came out, and I'm trying to think of the last time I wore that tie.
And you have a few months of beard growth on you.
Yeah.
Looking very good now. So Matt, I know this is an area that you truly are a world expert in. And the Association of Professionals in Infection Control and Epidemiology has said that clothing is a virtual conveyor belt for infections. And that's a very visual image. Can you tell us kind of what your experience and the data that you've worked with and kind of what you've learned about how apparel acquires, retains, transmits germs?
Sure. Apparel is such an interesting, we call them fomites in the medical community, but it's such an interesting surface to work with in many ways. You think about it, you get, when you talk about disease transmission, you can get it from the outside world, contaminating the uniform of a healthcare provider, but the healthcare provider themselves can also contaminate their own uniform, both from the inside out and also with their own hands. So it's a really, if you think of all the surfaces that we have really anywhere, apparel is by far one of the most dynamic surfaces that are in the world. And then if you just think about all the environments that apparel will be in, especially in healthcare on any given day, healthcare worker will wear it to the bathroom, they'll wear it to the locker room, they'll wear it to one patient room in one unit, go to another unit wearing the same apparel. So you can imagine that if it gets contaminated in one area, you're really dragging that contamination with you wherever you go. And by just the virtue of who we are as human beings, constantly touching different parts of the body, especially things like the breast pocket, our sleeves, and things like that, it's a very dynamic surface. And it really is a very complex thing. And we've done a lot of work with antimicrobial apparel. And one of the confounding issues that we've always had when we try to do any kind of clinical trial is that you're not just wearing one garment, you're wearing multiple garments at the same time. So if you think of a scrub top, you see nurses in the hospital. How often do you see a nurse wearing just a scrub top? They're almost always wearing a long sleeve shirt underneath, or they're wearing a fleece over the top of it, or a sweater over the top of it. So it's a really complex environment that our healthcare apparel lives in.
Yeah, for sure. Now, the other confounding factor there is, you kind of mentioned it, is it's easy to wash your hands after you see a patient. It's easy to clean a surface. Those things, are easy to clean and they don't move around that much, but the apparel is moving around very rapidly and you can't clean it very easily in between patient contact and stuff as well. So what about, specifically what everybody's worried about right now is human coronavirus on apparel and some of the research in that area.
Yeah, so this is a study by, at the bottom, referenced it's by Katie Laird in England. And she's just done a tremendous job of looking at pathogens on textiles and how they survive both industrial and commercial laundry. So she's really an amazing expert on this particular topic. And, if you look at the one in the middle is very clearly 100% cotton. I don't know what the one on the left is, but it might be a 100% synthetic. And then the one on the right looks like it could be a 60-40 polycotton, right? So they have seen some differential persistence of SARS-CoV-2, which of course is the virus that causes COVID-19. And that differential persistence, it also continues on in different types of washing, whether it's industrial washing or home laundry. We definitely see persistence of COVID or of SARS-CoV-2, but it's very interesting as how it relates to the different fabrics.
Yeah, I think that's one of the real thing, not just the fabric, but also the wash method. In industrial laundry and get those high temperatures. At home laundering, you can't get up to 140 degrees to really reduce things. So let's talk a little bit about, workers at risk. We know that different workers, and you talked a lot about the healthcare environment, but and we know that they always have been exposed to a lot of different contamination. But how does the apparel become contaminated? And not just healthcare, but in all environments, whether it's a firefighter, a policeman, someone wearing just general workwear in food service, say,
Right.
How would their apparel become contaminated?
Right. So there are three modes of transmission for pathogens. The first one here is fluid exposure. That's kind of a bulk transfer of fluid, an accidental splatter or a spill. You can imagine a healthcare worker in particular getting a blood splatter or a urine splatter or even vomit. You just look at really the second one, aerosol, sneeze or cough. That applies to all of us, where somebody who's walking by you or is right in front of you, or maybe even yourself, sneezes or coughs and jettisons that pathogen and all of that particulate matter, and it falls on your clothing. Contaminated surfaces are probably the most common for all of us. That contact transmission, where we touch it with our hands, or someone else touches our clothing with their contaminated hands, or we brush up against a surface with contamination on it.
Yeah, it always amazes me when you see the high-speed videos of someone sneezing and how far those large droplets will go out into the area. And,
yeah, if you can get over how gross it is, it is just an amazingly beautiful system that we have to jettison things from our,
Right.
from our lungs and from our throats. But I forget, there was a movie with Martin Short. No, not Martin Short. I forget his name now, but Inner Space. And I think they said, sneezes can clock up to like 275 miles an hour, something crazy like that.
Right.
I'm probably totally missing that, but
Yeah. So I really had a great fondness when I saw this study from the Occupational Information Network, looking at who the workers are at risk and kind of the paradigm they came up with. And they kind of made it, 3 buckets. One is workers that have known exposure, like a healthcare worker you were mentioning, where they're dealing with patients that are known to be infected, whether it's SARS-CoV-2 or methicillin-resistant staph or any other organism. And that's really how often are they being put in that infectious environment. Healthcare is a great example. The other big bucket, or the second big bucket, is unknown exposures. And these are jobs where you may be in contact with a lot of people, but you don't know the etiology, the epidemiology of those patients, not necessarily.
Right.
Transportation workers, like someone, a flight attendant on an airplane is locked in there with 100 and some odd people. She has no idea if they're contaminated, and she's in an aluminum tube, with 4 or 5 different groups of people all day long. A bus driver is another great example. TSA employees that are screening, hundreds of people all day long. They don't know what the infectious origins of those patients are, and they're coming in contact with a lot of them. And then the third is how close you're working with people, massage therapists, dental hygienists, physical therapists, people who are in close contact working with people, also are a great risk. And they looked at 966 jobs and rated the risk of these jobs. And what we did was take the top 100 jobs, who are the people, the top 10% of the people at greatest risk, and what occupations do they have? And healthcare was far and away number 1, personal care, massage therapists, physical therapists, people, medical assistants. Third was transportation. In fact, if you looked at the top 100, number 41 was flight attendants. Everything else before then was healthcare related. First responders, firefighters, police, EMS, education. A lot of kids all throughout the day locked in a room. Entertainment, interestingly. You have a lot of people in an audience of unknown etiology, and then food and food service. And that was how we rated risk. But the other part was, okay, well, those are the people at risk, or those are the people actually getting infections. And the advisory board group, if people aren't familiar with it, is one of the best researchers I've ever seen. The work they do is phenomenal, on population health and a lot of other medical areas. And they looked at the people actually getting infections. And I thought this was really interesting. Food and agriculture was number 1, and transportation number 2, as far as people actually getting infections. And then you can see down the list, health and emergency workers, which I thought was telling, because it really kind of speaks to that known unknown. The people that in healthcare can anticipate risk and they can wear personal protective equipment, whereas transportation, food service people, it's really hard to figure out what your risk is or prepare properly.
right.
Matt, what has been your experience kind of in worker risk in those areas, and thoughts on this data?
Yeah, I think it makes a lot of sense. I mean, maybe first blush you're surprised by it, but then once you start to dig into it a little bit, it makes a lot of sense. I mean, I think of my own situation. I'm a laboratory scientist. We're a microbiology laboratory. We know what our exposure is, and we control every aspect of that exposure. So our risk is zero. I mean, it's just above zero. You think of healthcare workers, especially nurses and physicians, they know what their risk is, but they don't always know, they can't control it, right? They can control it to some extent with PPE. You look at food and agriculture, they don't know what their risk is. They don't have any control over it. Sometimes in the food industry, you'll see there are sneeze guards and things like that in place, or somebody's wearing a face shield. But that's kind of rare, and they're face to face oftentimes with the folks who are bringing bringing the pathogen into their work environment. And you just look at transportation, and I guess is logistics like truck drivers and things like that?
I don't know specifically
Right.
how they defined it there, but transportation was flight attendants, bus drivers, people like that. I would imagine logistics workers would be bus drivers and stuff.
If you have contact with a couple hundred people a day, that's probably about what you're looking at. But they're literally running into thousands upon thousands of people every single day, coming within a foot to a couple of feet of that person's, breathing and things like that. So yeah, I mean, it makes a lot of sense.
Yeah, like I said, I was first surprised, but then it made a lot more sense when I looked at it.
Sure.
So let's talk about some of the gaps in healthcare worker apparel. The apparel that people wear, today is non-protective work apparel. And I love this picture because it shows, nurses back in the days of the plague with just regular non-protective apparel. And then you have the statue of the physician that had bubonic plague gear on. And, he has that day's version of PPE. The conical nose piece was a way to keep both distance, and they put incense in there. In their mind, they were killing the evil humors coming from the infected patient, as well as the breathing, like a mask. If you think of the strategy we have today, it really hasn't changed, right? We're doing the same exact thing a couple hundred years later. People wear non-protective apparel, and then when they anticipate exposure, they wear PPE. And there's kind of a gap there, right? I mean, it doesn't make sense that in today's world, we're using a strategy from the 1500s. And then, PPE, while important, clearly has a role. Sometimes people don't wear it. Sometimes you don't have it. You can think of a firefighter or a policeman showing up to an accident scene. They're going to jump in and try to save a life. I don't think they're going to think, well, where's my PPE? That isn't how they're wired. Sometimes it's worn improperly. And I thought this study was really fascinating. In the first SARS outbreak, 44% of the cases in healthcare workers occurred because they took off their PPE improperly.
Right.
That's just a staggering number to me, and it's happening today.
Yeah, and as we talked about the other day, it was a critical aspect of transmission in Africa during the Ebola outbreak, several years ago.
Right, and a very blood-rich environment, disease as well. So PPE, while important, doesn't always solve the problem and can actually cross-contaminate. So Matt, let's talk about some of the gaps in testing. Clearly, we need new strategies for worker apparel to protect workers, especially in settings where you have these unknown exposures and it's difficult or impossible to anticipate exposure to intermittent or unexpected exposure. You've worked with a lot of fabrics, a lot of different test methods and stuff. Kind of how can we link the lab effectiveness of these antimicrobial technologies to the clinical effectiveness of them?
Right. It's tough. I mean, if you might want to go to the next slide. These are the methods that are standard test methods that are currently out there in the world for testing antimicrobial technologies on fabrics. This survey was done by the RA75 group that I'm a part of with AATCC. And what we found, this is, the people surveyed were manufacturers of textiles, technology owners of antimicrobial technologies, and academic and commercial testing laboratories. And the question was about, what is your method of choice, right? What do you use most often to test antimicrobial textiles? And overwhelmingly, almost 90% of the responders indicated AATCC TM100, which is, I believe it's the oldest antimicrobial textile test method. It was developed in the 60s by AATCC, which again is a textile testing standard test method organization. The second two, we'll just say they're essentially the same because they're very close, are AATCC TM147 and ASTM E2149. What's really interesting to me about this is that neither one of those methods are particularly useful in testing antimicrobial technologies. TM147 also has the name of the parallel streak method. It is a qualitative test. You either have growth or you don't have growth of bacteria underneath the textile. And again, this is an old test method. I don't have the exact time when it was developed, but it's well over 30 years old. And it was basically designed to show within 24 hours whether or not your antimicrobial was present or not. Now, the antimicrobials that were used in the 70s, 60s, 70s, and probably the early 80s, those were absolutely leaching technologies, massive leaching out into the environment. You think of canvas materials that are used as tents that were designed to reduce things like mildew, and they had to leach out in order to kill an overwhelming amount of contamination. Well, we don't use those technologies very much anymore, if at all. And modern technologies are what we call, micro leaching technologies. They release a little bit of antimicrobial into the close-in fluid environment in order to interact with bacteria and fungi and now viruses. But they don't really leach out into, in this case, agar, which is the base material for this test. So it's a totally inappropriate test. It's hardly useful with anything anymore. ASTM E2149 is the wrist action shake flask method. That was designed by the developers of Aegis, which is a quat silane. If you don't know, quat silanes, when they're incorporated into just about anything, lend a hydrophobic barrier to that textile in particular, but really any surface that it's incorporated into. So it has a fluid barrier. Well, TM100 requires an immersion into fluid. So if it's requiring that kind of contact, you have to overcome that in order to expose the antimicrobial to the bacteria. And so the shake flask method uses a wrist action shaker in order to keep continuous contact between the antimicrobial and the inoculum. It does have a leaching component and a non-leaching component to that test. Highly impractical. The results within a laboratory are highly variable, just because there are a lot of things that can go wrong with this method. And the people who developed it have largely abandoned it as well. So it's still used, but obviously it's used a lot more than it should be. And the bottom line is, in all of this, is when you're looking to evaluate whether an antimicrobial is present or not, TM100 is a pretty good method. TM147 is not a very good method anymore. E2149 is also not a very good method anymore, and yet still 50% of the time people say that that's their preferred method, or not 50% of the time, but 50% of the respondents indicated that they do use those frequently. So they're clearly used appropriately. So you talk about gaps in test methodologies. That's the biggest one right there. People using the wrong test methodology for their antimicrobial technology. And the other one that we're going to get into in a little bit is the different types, the different tiers of testing that can be done. So let's see the next slide.
Yeah, so real quick before you move on,
Okay.
what would you use, or how would you use those methods? I mean, what do those methods tell us? You know, those currently available methods.
Yeah, so TM100, there's another one that's very similar to it called ASTM 3160, sorry, ASTM E3160, ISO 20743, JIS L1907. These are all very similar quantitative methods. They are 24-hour methods. And so basically what they're saying is, over the course of 24 hours, does your antimicrobial technology reduce the growth of bacteria? And they tell you each method is a little bit different. So they tell you slightly different things, but for the most part, they just tell you that your antimicrobial is effective at reducing bacteria over 24 hours in a liquid inoculum. Gotcha. Like I said before,
for the purpose of telling you if the antimicrobial is there on the
present. Yeah, and it gives you a rough idea. So when we incorporate antimicrobials into textiles, there's a lot that has to be considered. If you talk to a manufacturer, they would say more antimicrobial, the better. You talk to a technology holder, they would say, well, you need to add this amount of antimicrobial in order for peak performance. And you talk to a testing laboratory, and we just kind of say, well, it depends. It depends on the construction of the fabric, how the antimicrobial was applied, what that antimicrobial is, what other things were added to it. And so it is helpful to have these test methods, these standard methods that allow us to sort of across the board evaluate the different types of technologies, antimicrobial technologies, combined with the different textile manufacturing methods. You name it. The construction of the textile itself is also very important.
Very good. So, you look at typical manufacturer laboratory test results for these methods, whether it's the TM100 or the E2149, every antimicrobial technology will have their data with these methods saying that they have great results, 99.9% for everything. In your opinion, does the lab results equal what you see in the clinical results that have been out there?
It's very rare that they correspond to each other. And this is a big problem that we're having in the field right now. It's a twofold problem. One, the laboratory testing that we're doing at the moment does not represent the end use of, if you're using just standard test methods, they do not represent end use of the textiles at all. But also, if you look at the clinical methods, very few of them are done in a way that's really geared towards the complexity of antimicrobial textiles and how they're used in the environments that they're in, which I was talking about earlier. So we don't really have much in the way of good clinical trials that are out there on antimicrobial textiles. And we don't really have a lot of predictive standard test methods either. So there's a big gap.
Sure. So let's talk about that gap. You have the laboratory testing, which, as you said, is about determining if the technology is present. You have obviously field clinical trials that tell you if the technology is effective. What's that middle gap look like, and help us understand it?
Right. So I was talking about a tiered system before. Our current laboratory standard test methods are what we call tier 1. Field and clinical trials are what we call tier 3. And this real world worst case simulation, which is a laboratory based test, would be tier 2. Tier 2 methods are intended to be predictive of tier 3 tier 3 trials. So they are using the controlled environment of the laboratory, but they're using scenarios and parameters that will help predict how a textile will perform out in the field. And these tend to be fairly complex. Right now, we don't have any standard test methods out there that fit this category. The only one that I'm aware of is one that we developed for Vestex many years ago now, which we call, the splatter challenges. And that is what's represented here. It's a worst case scenario that the effort is to correlate with clinical testing. We're using inoculums that are seen in the clinical setting. If you look at TM147 and methods like it, they immerse the fabric in a liquid inoculum, which is not seen out in the real world. So instead of doing that, we're inoculating using those splatter that we talked about before, an aerosol or direct contact method. And this allows us to see how the fabric itself interacts with those types of transmissions. And we're using typical healthcare pathogens in relevant inoculum sizes, right? And so the time points are also relevant. The nice thing about this is we've presented this to the FDA, and we have gotten acceptance of it through the FDA.
Very cool. So talk about a little bit of the results that you've seen and how that differentiates.
Right. So this is an interesting one. This is from the original fabric challenges that I was talking about. This is the liquids or the splatter challenge. And we see an untreated control, which is just a 60-30 or a 65-35 polycotton without a fluid barrier technology or chemistry. And then there's Vestex, which, Ben, you'd have to correct me, but I think that was a 65-35 polycotton.
Correct.
With an quat antimicrobial and fluid barrier chemistry as well. And we tested at 0, 15 minutes, 30 minutes, and an hour. And the most interesting thing to me was right off the bat, we had a 2 log reduction on the Vestex, which, that's not enough time for an antimicrobial to act. So that's clearly showing you the value of a fluid barrier on the textile. That's a 99% reduction right off the bat as soon as it's inoculated. And then we see the steady progression downward where we go, from a 2 log reduction at time 0 to a 3 log reduction at 15 minutes, roughly the same at 30 minutes, and then down to a 4 log reduction at an hour. And so you're seeing the impact of the antimicrobial as well as that fluid barrier once you get out to an hour.
Very good. So as far as a correlation testing, there's been a lot of work in the laboratory for other technologies, and then, Vestex has been able to show success in the clinic as well as in the simulation environment. Do you know anyone else that's done simulation type work?
No, there are a couple of groups that are working towards it. I don't think that they've been as successful as we have with it. I think their methods tend to fall short of actual simulation. I do know that anybody who works in the field of antimicrobial wound dressings, the FDA requires a very rigorous simulation test there. That's the one area that has, and it hasn't been developed into a standard test method to my knowledge, but that's another area where we've seen a lot of activity. But insofar as apparel goes, no, I haven't seen anything other than what we've done.
Yeah. So Matt, what are the
can you go back to that real quickly? I think it highlights a really important thing here. So if you look at the top two, the single mechanism silver, the single mechanism chitosan, we see that, they've got, the first one has decent reduction of Staph aureus. It's not, 95% is not bad. They're using TM100. That's a 24-hour test method. So that's actually, I would argue, not particularly good, actually. An 81% reduction over 24 hours is essentially a non-existent. That's very hard to quantify. So I would argue that that's not a reduction at all. So then you look at the clinical trial, And we don't see any difference between conventional and antimicrobial scrubs. The only fabric that's shown laboratory effectiveness and simulation effectiveness as well as clinical trial effectiveness is the Vestex that we see here. And I think largely, when we look at that, we are able to use a standard test method like E2149 to show that this is effective. We do that with a simulation approach, and we also show that it's super effective for log reduction. But here's where we get to something very interesting. The Bierman study is one of the better, if not the best, of the antimicrobial textile clinical trials out there. And it's the one that shows a reduction, a strong reduction on antimicrobials versus conventional scrubs. And I would argue that the other clinical trials, certainly the ones that I've reviewed, other than the Bierman study, just are not designed to show effectiveness of an antimicrobial textile. I just don't think the studies are well designed.
Yeah.
So it's a super important part of this whole thing. We can't base our decisions off of clinical trials unless that clinical trial was well done.
Agreed. Totally agree. So Matt, there's a lot of discussion about what's the regulatory implications of all this stuff, and how does that correlate to what you can actually say for promotional purposes. So can you kind of walk us through the difference between the FDA, the EPA, and what that all means?
Yeah, so I have more experience with the FDA, more personal experience, I should say, with the FDA. And the FDA 510K is a process that is based off of valid scientific evidence. You really, when you're presenting to the FDA in an area where they're not seeing a lot of products. Again, if we go to antimicrobial wound dressings, they probably see, 100 to 200 of those a year. They know what they're looking for. There's no wiggle room. You have to do exactly what they want. That's rote. When we're looking at antimicrobial apparel, there's very little out there. There's your 510K, and I don't think there's anything else at the moment. Correct me if I'm wrong.
Not that I'm aware of. Vestex
Yeah.
is the only FDA cleared antimicrobial fabric I'm aware of.
And so when we did this, it was the Wild West, and we had a lot of arguments about it. But it was the kind of thing where we knew that if we presented the best scientific evidence that we could with exquisite rationale, meaning that every decision we made was based either on science or medicine or any other kind of data that we could find to justify our protocols, we had to provide all of that. And as long as we did that, we were fairly certain that we were going to have a successful submission. Now, contrast that with the EPA, and the EPA is far more, I used the term rote before about the EPA and antimicrobial wound dressings. EPA does that with just about everything, right? So they like to have firm protocols that are the same from one product to the next, to the next, to the next. In the pesticide world, which is what EPA refers to antimicrobials as, so in the pesticide world, they're largely looking at, their biggest database there is liquid disinfectants. There are a lot of agricultural pesticides as well, but they're a little less applicable than liquid disinfectants. And EPA has stayed out of the textile game because they have a clause in there where in FIFRA, you can claim a treated article exemption. And the treated article exemption is that you can use and state that you have antimicrobial chemistries on your textile as long as it's there to protect the textile. And that protection includes odor reduction as well as inhibition of molds. Now, they have really stayed away, everybody has stayed away from making anything other than treated article exemption claims in the antimicrobial textile world. I believe there's one group out there that has gone after specific bacterial reduction claims as an antimicrobial textile, but it's very, very rare. So much so that the EPA kind of doesn't know what to do with it. But when it comes to, we're in the middle of a pandemic and hopefully near the end of it, this treated article exemption doesn't apply to viruses. And of course, everybody with an antimicrobial technology in a textile wants to make an antiviral claim, and as they should want to. So because viruses do not have a negative impact on textiles or whatever the treated article is, you can't make that treated article exemption claim. So now the EPA is going to have to weigh in on anybody who wants to make an antiviral surface claim, whether it's a solid surface or a porous surface or even a soft, non-porous textile. If you want to make an antiviral claim, you are going to have to go through the EPA regulation process. So the problem there is we don't know what that process will be. And I haven't seen any guidance on that whatsoever.
Yeah, so if you're wanting to make just basically odor, mold, and mildew claims, you can just do the basic laboratory testing. That's going to tell you its effect, if it's there or not.
Right.
If you want to do real world laboratory testing for viral claims, then maybe you have a route with the EPA, maybe not, or with the FDA. Clearly, that will be required. And then clinical testing for the FDA as well. Would that, as far as the testing that would need to be done for those claims, be accurate?
Yeah, and again, I'm hoping that we start to see a little more clarity from the EPA as to what they're going to require. The FDA, again, I think they've been, we had a big snafu with the emergency use authorization for decontamination of face masks. That was a difficult one that the FDA put out there. Very few companies were able to get their emergency use authorizations through the FDA on mask reuse because the guidance was very, very different from one group to the next. But now that we're sort of away from that, the FDA seems to be calming down a little bit and using a much more thoughtful approach to antiviral claims with treated textiles.
Agreed. So what does that kind of translate into real world? And Matt's an independent lab and stuff, and this is a slide specific to Vestex. And Matt has helped us with our FDA filing, and his lab did a lot of our work, but he is a totally separate independent company. But I just wanted to share with everybody kind of, a compare and contrast brands that license Vestex and are protected by Vestex, what they can say versus the odor and inhibit mold and mildew claims that all the other technologies have to say. So a Vestex designed to protect personnel from transfer of microorganisms, body fluids, particulate material, provides protection from intermittent unexpected exposures to microorganisms. Actually, Vestex has been shown to reduce blood, urine, sputum, gastric fluid, and bleach, and that's part of our FDA claims. Reduces 99% of MRSA, and we have a pending claim with human coronavirus in under 1 minute. Proven in the clinical environment to reduce methicillin-resistant staph 99.9%. That dual mechanism of action between the fluid repellent and the antimicrobial has been shown to not only reduce the bodily fluids, but also prevent colonization of methicillin-resistant staph. And then Vestex is also endorsed by the American Hospital Association, recommended by the National Science Foundation, was selected as one of the best infection prevention products by Newsweek, as well as endorsed by Clinicians Report in the dental market. So that gives you a good comparison and contrast between a technology that's gone through the rigor and testing, both in the simulated real world environment, as well as the clinical environment, such as Vestex and the claims we're allowed to make with Vestex versus all the other technologies out there. And what we do is we license Vestex to other brands who want products protected by Vestex. We work with them on developing products that are protected by Vestex, and selling those products to consumers. And then we work on developing brand insistence for consumers and businesses who today want to be protected, unlike the non-protective apparel that people are wearing today. Take-home messages. Clearly, worker protection is a top-of-mind issue, not just for workers, but in the C-suite as well. Germs rapidly acquired, retained, and transmitted on healthcare worker apparel. And wearing non-protective apparel is an outdated strategy. We can't continue to use strategies from the 1500s in today's new world. PP only works if it's available and if it's worn correctly. The Vestex Fabric Challenge is the only method that has been shown to correlate between both the tier 1 testing that shows the antimicrobials present and then creates that correlation to the clinical testing. And then if you want to make claims about effectiveness and safety and the proper testing to expand your promotional claims to address these public health emergencies, you need to do the proper testing to get your products approved appropriately. Matt, anything else from your end?
Yeah, I would say real quickly that in the last 5 years, we've seen a real groundswell of support for better testing of antimicrobial textiles. When I first started in this area a little over 10 years ago, I was incredibly dismayed to see the lack of scientific rigor, the lack of activity in the development of standard test methods. And while I appreciate the folks that came before, I just was very disappointed that there had been so little movement in really the last 60 years. And there was no movement at the time with regards to that tier 2 testing that we've talked about, the simulation testing. And so what we've seen in the last 5 years is sort of a revitalized testing community, re-engagement of testing with, from the manufacturers and the technology holders, as well, in fact, with the end users. So people are demanding higher quality testing. They're not just going for the cheapest test method that'll tell you whatever you want. They're going for test methods that give data that they can use both in product development as well as in marketing material, and now finally in regulation of their products. And this has been the work of many groups, including our own. Ben has been a big proponent of this as well, and I appreciate that. I think if we had not done this, we would not have been able to respond as strongly as we have as an industry to the pandemic.
Right.
The pandemic really did need antimicrobial textiles for a variety of reasons. We're going to have masks with us for a long time now, and having masks that protect other than just stopping particulate matter, but can actually kill bacteria, fungi, and viruses. I mean, that's going to be a big comfort to people. And we're now able to do that testing and show effectiveness of a variety of textiles against viruses, bacteria, and fungi. So it's a good time to be a part of this. And I'm really proud of the work that we're all doing. So,
yeah, well done. Well said. Thank you. So with that, we'll open it up for questions. Steve, any questions?
Thank you guys. That was a lot of information. So, in my naive way, it feels like a tier 3 test with people going to a work site, grabbing textiles, like at a change of a shift, right? Or, just randomly sampling, and then bringing that to a lab and saying, okay, well, here's, these random samples that were performed. Is that just not happening at all? Or am I misunderstanding maybe what a tier 3, ideally, it seems to me, Ben or Matt, that's what a tier 3 should be, right? Is grabbing things from out in the wild and then testing it.
Yeah, I mean, tier three is classically more of what we refer to as clinical or field trials, and that's where you've got people in with regards to an antimicrobial textile, they're actually wearing the textile, and your endpoints are going to be anything from odor production to decontam or bacterial contamination to viral contamination is not so easy to do, all the way out to reduction of healthcare acquired infections. So those are clinical. Those are the tier three test methods. What you're referring to is more of what we call environmental sampling, and environmental sampling is valid. I would say that the results are fairly applicable to real world use because you're taking things from real world settings, but then you're putting them in the laboratory and testing them. So it's a valid way of doing. It's not quite a clinical trial, not quite a field trial, but at the same time, it's better than most laboratory trials would be in showing
really?
how things are
Okay.
performing in the real
Yes,
world.
Steve, what I would say for validating a cleaning process, a washing process, what you're talking about is an interesting idea. And it may show a difference between a non-protected textile and a protected textile with Vestex or another technology, that if it's just sitting there. But as Matt said, what we looked at was one group of nurses wearing Vestex, one group wearing regular non-protective apparel, and then every 2 weeks they would cross over, and the group that was wearing one was wearing the other, and every day they would culture the apparel to see, both at the beginning of the shift and at the end of the shift, to see what was on the apparel. And what we saw with Vestex was a 99.9% reduction both at the beginning of the shift, which is important because it means the contamination on the clothing isn't going from room to room to room around the hospital or for patient to patient to patient. And then at the end of the shift is important because that means the contamination isn't going out to the grocery store or home to the family on the healthcare workers' apparel. So that's how we studied it. And we were able to show a highly statistically significant reduction at both those time points, which was really important to the FDA and as well as the researchers. I mean, it was published in the premier journal in infection prevention.
Mm-hmm. Mm-hmm.
So a lot of the focus, is on the medical industries, if you will, Ben. But it occurs to me that the cliche of every outbreak movie, is blood from animals that
Right.
has wound its way into human worlds, as well as it occurs to me that last year we had all of that news, what was happening at the meat packing facilities. And,
right.
Yes, that's because people are standing too close to each other, but it's also because the cliche again is that they're smothered, in textiles that are smeared with blood. It's some human, mostly animal. Have you seen applications for this or discussions in more of industrial settings than medical settings?
Yeah, so you make a great point. When we originally developed Vestex, the problem was in the healthcare environment, right? It was healthcare workers that were being, acquiring, retaining, becoming colonized, and getting infections with methicillin-resistant staph as the primary organism on healthcare worker apparel. But today, that's changed. We had applications where you had workers being with exposure in food processing, food manufacturing and stuff, but it wasn't as critical and there wasn't as much health risk to the workers. Well, now with COVID, that's all changed.
Yeah.
So I think the same premise now in healthcare now applies to flight attendants, transportation workers, EMS, firefighters, prison guards, all these settings where people are in close contact with people of unknown etiology, where it's really hard to anticipate, and you have these unintentional or accidental exposures to whether it be bodily fluids or an aerosol or direct contact with something, is kind of the next frontier for Vestex for sure. Matt, any thoughts on that?
I'm sorry, cut out there for a moment.
And any thoughts on kind of where the problem of antimicrobial textiles, it kind of started in healthcare, but where it transitions to next, or other markets where this is becoming more of a problem?
Yeah, I do think that, we need to be very thoughtful and engineer our antimicrobial textiles for the environment they're going to be in. When you talk about like a meatpacking facility, you're getting a lot of foreign organic matter on your garment. Having a fluid barrier there where you can just wipe that off and not have any residual organic soil, that will greatly reduce the chance of a virus or a bacteria, whatever the pathogen is, sticking to your uniform and actually having a food source, right? So viruses, that's not a problem, but with bacteria, blood or other organic matter can certainly be a food source. With viruses, that blood or organic matter can act as a stabilizer for the virus so that it remains active when you leave and take it off and accidentally ingest it or breathe it in. So we need to be very thoughtful about how we do these things. I'm not one for, let's apply the best antimicrobial to everything and that'll solve all of our problems. That's not going to work.
No.
Some antimicrobials will work in different settings than others. Some, we just absolutely require having a fluid barrier for certain things. And then we start to get into the specifics of uniforms anyway. We can apply anti-odor technologies to under the arms and things like that, while applying, fluid barriers to the torso area. And there are any number of ways of doing this. Or really targeting sleeve cuffs with an antimicrobial, because we know that we touch our sleeve cuffs all the time. So we just, as we get into this a little bit more, we see it right now. Textiles are becoming so sophisticated that everybody needs to be working together to really take the technology that we have and apply it in the best way.
Totally agree.
Yeah, I like that. I like that answer. Yeah, well, gentlemen, thank you so much for the education, and we've recorded this, so we will post it and spread the word, and get some more believers. And hopefully, as the two of you were saying, I'm encouraged that perhaps we have more bodies and regulatory bodies taking a look and issuing more minimum standards, right? I mean, look at, it did take, kind of an ANSI a long time to, get certain things in place for certain worker related, safety precautions and items. And, I'm hoping that this, the bodies that you were citing, and, that all continues. And it certainly helps with the good work you two are doing.
Absolutely.
Thank you.
Thanks, everyone.
Appreciate it.
Thanks. Take care.